Very good evening, everyone. Please confirm if you all can hear me. So I hope you all are excited for today's session. Very good evening. So everyone, I'm Dr. Tanushree and I welcome you all to today's live Q&A session on careers in AI-powered chemistry, cheminformatics, drug discovery, and the future. We are delighted to have you all join us for this interactive session where cutting-edge science meets real-world career pathways. As we all know, AI is rapidly transforming chemistry and drug discovery, reshaping how molecules are designed, how data is interpreted, and how future scientists build impactful careers. So, whether you are a student, researcher, or professional looking to explore AI driven chemistry, you are in the right place. This live QA session is a chance to hear directly from our experts who are shaping this dynamic field. Today's webinar is designed to be practical, insightful, and interactive, where our experts will address your questions on career opportunities, required skill sets, industry expectations, and how one can successfully transition into AI-driven roles in chemistry and pharmaceutical research. So before we begin today's live QA session, let me take a moment to introduce our experts who bring deep expertise to today's session, to today's discussion. We have with us Mr. Vinayak Khairnar, who is an independent chemistry consultant with 24 years in pharmaceutical R&D, specializing in medicinal and computational chemistry, process development, and cheminformatics. Formerly with Origin Oncology, he advanced multiple oncology programs, delivering 4 preclinical and 1 clinical candidate. Mr. Vinayak integrates AI-driven drug design, digital automation, and competitive intelligence to accelerate discovery, optimize processes, and strategically position innovation in a global pharma landscape. We welcome you, sir.
Thank you.
Also, we have with us Mr. Prodyot Banerjee, who is an accomplished bioinformatician and computational biologist with an MTech in bioinformatics from Delhi Technological University and is a qualifier of national level competitive exams like GATE and CSIR NET. Mr. Prodyot specializes in computer-aided drug designing, including virtual screening, pharmacophore modeling, molecular docking, and MD simulations, along with advanced genomics data analysis, such as phylogenetics and GWAS. Mr. Prodyot's scientific journey is enriched by a rich tapestry of research experience across India's premier institutions, and his work has been published in high-impact SCI-indexed peer-reviewed journals. Currently, he's working as CRO scientist, CADD, bioinformatics, and genomic specialist at Biotechnica. We welcome you, sir. Also joining us today, Dr. Snigdha Tiwari, who has recently completed her PhD in computational biology at the Translational Bioinformatics Lab, IIT Roorkee. She brings a rich academic foundation with a background in chemistry and bioinformatics, alongside a passion for interdisciplinary research and advancing biotechnology or healthcare through innovation and collaboration. Dr. Snigdha has made significant contributions at the intersection of basic and applied research, particularly in protein-protein and protein-ligand interactions, molecular docking and simulation studies, and healthcare or clinical data management. She currently serves as an academic support specialist in the Bio-IT and Clinical Research Department in Biotechnica. We welcome you, ma'am. So we warmly welcome ma'am and sir, and are truly honored to have you all with us today. Dear attendees, welcome, sir. So, dear attendees, we encourage you to actively participate and drop your questions in the chat section as we go along. As you all know, it's a live QA session, so we really appreciate if you post your questions. If you have any questions related to AI in chemistry, cheminformatics, AI in drug discovery, please feel free to drop your questions in the chat. So with that, let's begin this exciting conversation on the future of AI-powered chemistry and drug discovery. So as we all know that this particular session is all about careers in AI-powered chemistry. So sir and ma'am, I have I'm very much curious to know, actually, to understand, especially not from chemistry background. I'm very much curious to understand that what exactly AI in chemistry means. So, what does AI in chemistry actually mean, and how it is different from traditional computational chemistry? Vinayak sir, would you like to answer this question?
Yes, sure. I'm very happy to answer this question. So now it's like the for many last 10 years, if you see, so AI is introduced. Now before that we just do the our chemistry or computational chemistry very traditional way. So if I think AI in a chemistry, it's about the machine learnings. It's a deep learning and generative models applied to the chemical problems. So instead of solving equations, AI learn directly from the data's chemical data's reaction, molecular structures, and assay. While the traditional computational chemistry is a physics base, it really need many simulations like quantum mechanics, molecular dynamics, also docking, and it's a rigorous and interpretable, but it's slows and limited to the small systems only. So key differences I can point out here is a computational chemistry is a physics driven, mechanistic, slow, but it's explainable. But while this AI driven chemistry, it's completely data driven. Without data, it cannot survive. It is a predictive as well, while computational chemistry is not much predictive. So it is a one to one kind of experimentation. It's a fast and able to generalize across a vast chemical space, while this computational chemistry doesn't have that chemical space. That season we say that it's a very slow, but it's a explain, but still it is an important tool. So for example, nowadays, earlier we need to calculate the reaction yields, for example, in a chemistry. So what we used to do, we need to do the experiment, then complete the synthesis, and then have some figures in this one. So it takes a lot of time, but computationally we can do this DFT simulations of the transition states. This AI learns the yields trend from the thousand of reactions. So here, not to do the experiment to one experiment, it just recognize the patterns, whatever the data is available from the thousand of reactions. So that way, AI helps here. Also, second example I would like to quote is there. Quote this protein ligand binding. So instead of simulating every interaction, now AI model is availability like that. It uses a deep dock kind of algorithms and predict binding in a binding poses in a seconds, where computational chemistry, traditional communication take the more time. So it's like that, very simple way I would like to explain this. This is the traditional method, like that, solving equation carefully on a paper. So when we get the any equation, we used to solve the equation on a paper. But this AI give the advantage like that, it is a calculator, but along with calculator, it also gives the next step based on the experience. So overall, AI chemistry doesn't replace the physics or chemistry or any science, but it accelerates it like the moving from a bicycle to the an e-bike. So where the destination is the same, but you get there faster with more power and with the ability to explore far more terrain. So that is the advantage of AI. So it is not replacing any kind of science or computational chemistry, from my perspective.
Thank you. Thank you so much, sir, for explaining in such a very simple and wonderful way with wonderful examples. Uh, even I would like to know that we are being, I have like we have heard about bioinformatics a lot, but now there is a term cheminformatics. So I'm very much curious to know, and I'm sure many of our attendees are very much curious to know that what is cheminformatics and why it is becoming so essential for modern chemists, especially like if we talk about both in research and industry. So Pradyot sir, would you like to explain like what is cheminformatics and why it is becoming so essential in research and industry?
Yeah, surely. First of all, thanks for having me. And Hello to all of the guests out there. So basically, when we are talking about cheminformatics, as the name suggests, chemistry and informatics. So it is an amalgamation between two of the subjects in an interdisciplinary field. So which collates two of the disciplines, basically chemistry and informatics. So just like how we have bioinformatics for biological data, and we are doing the analysis using informatic tools or basically AI-driven tools, similar to that, if we are doing the same thing in chemistry, so that would be referred to as cheminformatics. And basically, taking the previous question in mind, so basically, what the AI is doing. So AI is basically automating all the things that we used to do manually. Okay. So as Sir has just mentioned about the diff doc, if I'm just taking the same example, okay. So we used to do docking in gold, then your Autodoc Vina, this that. Okay, so there were multiple restrictions to that, that we need a Linux system. So then it is time taking. Apart from that, if we go for a blind docking, it was computationally very expensive. The reason being we have to like look for all of the contours of the protein, okay, cavities and the pockets. Now that particular restriction is not there because we are using a cloud server and all the things are automated. We'll be using a Python based API, and that will solve the problem for us. Okay, so and it will be done in seconds. Okay, seconds to minutes. It will be automated. We don't have to select any what do you say, your arbitrary box that we set. Apart from that, all of the things, the exhaustiveness we can like increase also. So that would be an example. Likewise, so that is basically about the chem informatics. So it is just an interdisciplinary subject which caters to both your chemistry, and in order to automate that particular chemistry, we are having the informatics based tools or web servers that can help us with catering to the problems of chemistry. Yeah.
Thank you. Thank
Thank
you
you.
so much, sir, for explaining so wonderfully about cheminformatics. Snigdha, ma'am, I would like to know from you, like Pradut sir mentioned about docking, that we are using AI in docking. So what are your views? Like how does AI can accelerate molecular design or reaction prediction and screening compared to classical methods which we use in uh, drug discovery process?
Yeah, sure. Good evening, everyone. Very nicely defined by Sir, like both of them. I would just extend what has been explained. Comparing to the traditional knowledge, we used to do force fields. We used to do like physical things, Newtons, atoms, molecules, and then develop the ligands in the in the lab setups and everything. It would take a lot of time, cost. Um, as the procedures are moving towards big data, we are moving towards big data, we are moving towards the analysis. We it's now mandatory to work with AI, ML, QSAR modeling, docking and simulations, right? So the AI would definitely help in all the prospects of cheminformatics. Starting from gathering the information, we need to gather the information, we need raw data. A lot of raw data would be needed for ML training, validation, testing sets, right? So starting with the collection of the data to processing it, getting the results, validating the result, and then getting the results, doing the hypothesis and getting the result. So from the collection to the result, to getting the endpoint and the result, AI actually plays a very important role. How would you collect the data? From the help of APIs, from the help of Python as a language, or R as a language or anything. The collection of a data using APIs, using AI and ML algorithms, and then doing the pre-processing of it. Cleaning is so important. Your data is so important. So pre-processing it, based on the AI, AI and ML. I mean, all the libraries, the deep chemistry. We are working with with the scikit-learn, TensorFlow, all of that, and then building the hypothesis, and then testing the result. What the accuracy is, what the confusion matrix is, right? So all the reaction predictions, if it's a kinetics, if it's whether it's a dynamics, whether it's the IC50, EC50 values, screening, talking about the screening, virtual screening, we do use a lot of tools now. Say for example, we use Autodoc Vina, we use Adme Labs, we use different kind of AI tools now, and highly highly cited, getting the high kind of accuracy. It is now mandatory to, for being, being it as a QSAR modeling, structure prediction, or as sir mentioned, we don't need to build the grids separately and waste our time. AI would do now. I mean, AI and ML is doing great job. So learning AI and ML, and then using it, starting from the collection to result, is mandatory now, I would say, is the need of an hour. Yeah.
Thank you. Thank you so much, Snigdha ma'am, for providing insights on cheminformatics and how AI can accelerate molecular design and other processes which are required in drug discovery. So, since now we are familiar with the basics of what AI in chemistry means and cheminformatics, so since you mentioned about tools, so moving over to the tools and technologies which can be used in AI in chemistry. So, many students are new to this particular subject, chem informatics. So, for students which are new to chem informatics, what are the core skills or tools you all can suggest that they should start learning first in order to make a career in this field? So, Vinayak sir, would you like to provide some insights on this, the tools
Yes,
and the skills?
sure, sure. So now we are very lucky to have the kind of new generation kids, we can say Gen Z and all. But when we start this, it is very old. Should we have to each and everything we have to scratch from the, even we don't have the internet at in those times, so we have to kind of softwares, and we have to completely depend upon. But there are now many softwares and tools are available online, and also very advanced tool also available. But just to start as a student learn this one, so I can suggest two or three what is the free open sources tools are there, like this RDKit, some kind of DeepGame or Autodoc. These are the very fundamental tools one beginner chemist should learn from the when they start their career, or they want to start any they want to do the PhD or something. This is a basic skill. For example, RDKit, this is the foundational library for the cheminformatics. So it handles like that molecular representation, like the smiles. Smiles is a kind of language that chemists talk with the kind of computer. So smiles is very very important. Also fingerprints also is very important. The it also helps us to substructure search, and it's also we need the many descriptors. It also helps generate the many descriptors whatever are required for your computational toolkit. So every chemist working with the AI should know the RD kits, and it's just dictionary of the molecular data, I can say. The second tool I can suggest, or everybody will agree on, so it's a deep chem. This is a higher level of library that integrated this RD kit with the this machine learning frameworks, like that. Now we use the TensorFlow or PyTorch. It provides ready to use models for the QSR docking and property predictions, like that. Many properties we predict toxicity, solubility, permeability. Think of it as a grammar book that teaches you how to write the new sentence with the yeah. And also third tool I can recommend, just to start with, this Autodoc, or we say just Autodoc. It's a classical docking software. Still it's a old, but still it's widely used, and it is a, I think it is a open source tool. While it is not AI first, it teaches the basics of ligand protein binding and scoring function, and this is a very very fundamentals for the any kind of computational chemical, and the skills that remain relevant when comparing AI driven docking. So I can say this just to learn with the start with the RD kit. And if chemistry is we can say it's a language, then RD kit is the kind of dictionary, and deep chem is the kind of grammar book, and that teaches how to write the new sentence with the yeah. This I can construct my answer. Hopefully everyone agree with this kind of this one, and some other thoughts also are welcome.
Thank you so much, sir. Pradyut sir, Snigdha ma'am, would you like to add some tools or skills?
Yeah, sure.
Yeah.
If I may start, sir. Yeah, so just extending what sir said, knowing your data is important. For me, data is everything. We say in machine learning, garbage in and garbage out. We just can't feed garbage to our machine learning and then, garbage is a result. What is important is, of course, the basics of database. From where will you collect the data? From where will you get the raw data to process, right? So you need databases to know very well, know your data, ligand data, your target, your protein data, what is a uniprot, what is the ncbi, what is the pdb, and why it is, I mean, how it is curated, what is the secondary database, what is the tertiary database, why it is called so, what are the integrations that that goes along with the uniprot, with with the databases, because you need the raw data, of course, for your model building or something. Even if you're working ligands, practical, if you're, I mean, collaborating with the practical ligands in the chemistry, you need to know your ligands, you need to know your data, what is it actually, what is the moiety, what is the chemical structures, and then after that, of course, the basics come in with the python as a basics. What are your data types, what are the variables, how the variables needs to be stored, how do you do the pre-processing again, and then for docking, definitely autodoc vina, autodoc, highly curated, I mean, highly I mean, it's an open source also. uh, so go along with autodoc vina, autodoc. I would highly encourage to do, for the validation of autodoc, because validation is important. Validation for your autodoc, just go ahead and learn gromax. Go ahead and learn your simulation. How would you do, how will you simulate, and how will you validate your data? That is very important. So all these basics, python basics, maybe R as a basic also, to develop your hypothesis, to develop your statistics. Start with a basic. I'm not terrifying you, but start with basics. Just to open your Google Colab and start writing something. Just open Autodoc and start with with very simpler databases, and and look onto your ligand. How it works, how 3D. Pymol is very important. To visualize. Start with Pymol, Chimera, Autodoc, Gromax, all of these. Yeah.
Thank you, man. It's a very good addition.
Yeah.
Yeah, thank you so much, Saran ma'am. Uh, I request all the attendees, please feel free to post your questions in the chat. The session is all about AI in chemistry, cheminformatics, and drug discovery. So if you have any questions, if you want to make a career in this field, and if you have any questions related to this particular field, we have our experts with us. Feel free to post your questions in the chat, and we'll be happy to answer them. So since we are talking about AI, there is a lot of debate about how reliable are AI tools. So Pradyot sir, I would like to know from you that how reliable are AI models, like when we talk about GNN or AlphaFold or DivDoc or diffusion models in real research scenario, how reliable they are? Like, are they truly making an impact in chemistry? I would like to know your views on this, sir.
Sure, sure. Of course, it is making a huge impact. So as you can see, if you go by the research papers also, so they are published in your Nature to Lancet. So like a very high impact factor paper they are published in. That is the first thing. Second thing, of course, when we are talking about the AI based model, we do validate that as well. Okay, so using your confusion metrics and what is the efficacy or the accuracy of that particular system that we look for. Mostly they score about 88, 90%, 94% of accuracy. That is a very good one. Okay, and when we are talking about from the likes of your AlphaFold particularly, so we can get to an accuracy of 98% or more. So basically, what is AlphaFold doing? Basically, it is predicting a particular protein structure about which we don't know using Evo transformers. So using those particular things as it is doing, so what, how we can validate that particular result? So it is published in the Nature. That particular paper is published in Nature. You can see that when we are overlapping a particular protein structure procured from the wet lab based experiment, and when we have predicted that particular very structure using AI based thing, so it is having 98 to 99% of similarity, not only at the periphery but also at the domain levels, because the domain does matter. Because when we are going for your docking or this, that we are looking for molecular interaction using your, let's suppose after post-docking, we look for the interaction using your Discovery Studio to the likes of your in Glide also you can see, or any of the visualization tool, for instance, your PIME, all this that. Okay, so there you would see that the domain, everything it is predicting very well. Okay, so of course it is very much reliable. And for all other kinds of analysis also, let's talk about the molecular generation per se. Okay, so the initial problems that we used to handle in the wet lab is nowadays, as you see that there are hybrid papers, we do integrate AI based, algorithm based things. Okay, so for the virtual screening and do the docking, and suppose that we validate that particular result using either in silico or your wet lab experiments. So, of course, that is a testimony that the things are going right. That is why in the validation method also we are getting the similar results. Okay, that is the first thing. Second thing, molecular generation. So, if we were to do it using a wet lab based experiment, getting the relevant lead molecules, lakhs of lead molecules at a time would have been very much challenging. Whereas that particular thing is very easier when we are building a model of AI and coming up with multiple of the lead generation using your GNN or to your any of the other what do you say, your models that we have. Okay, so by training the model using already available chemical databases such as your PubChem to the drug bank to the your Bitwiz, Hugging Face, this that. Okay, so yeah, that's all. Thank you.
Thank you. Thank you so much, sir, for such a wonderful explanation. We have few questions in the chat. I would like to take them up now. So we have a question from Sri Rama Murthy. The question is that, sir, can you please explain hit to lead QSAR SAR analysis, scaffold hopping in designing small molecules NCEs? Vinayak, sir, would you like to take up this question?
Maybe I recommend Doctor to Murthy to
Okay.
answer this.
Okay. Pradyot, sir, Snigdha, ma'am, any one of
Sure.
you would like to take up this question?
May I?
Oh, yeah. Yes, sir. Please go ahead. Please go ahead, sir.
Yes. So basically, heat like a hit to lead, or let's first understand what is a QSAR and what is SAR first of all. Okay, so basically, whenever we are trying to, I'm just trying to simplify the concept. I'm not getting onto the details of it. Okay, so I'm just trying to simplify in a layman language. So basically, so basically, SARS or QSAR is basically a mathematical model which basically correlates between the structure of the chemical molecule and what are the functionalities that it may be concerning to or basically relates to. So based on the QSAR modeling, what we can do is based on the chemical structure, we can predict that what can be the features that it has, like the bioavailability, how we can increase that, like which particular group, if we change in the structure, so that will increase the bioavailability of that particular substance inside the body, or if it is being highly toxic, like if it is not passing any either of the ADMET parameters, like the pharmacokinetics and pharmacodynamic parameters. So what are the changes that we can do to the structure of the molecule, so that basically what we are doing is we are basically modeling that particular thing, okay, or manipulating the structure of it to make it work in our way, okay, or like in a better way, how we can make it more of a molecule that we are like targeting. Okay, so that is about the QSAR modeling. And hit to lead basically, like from the lead library we want that maximum of the hits we are getting. Okay, and based on that, if we can do the QSAR based thing, so like whatever be our intention to get a particular molecule about, like let's suppose I want a molecule which is having very high like bioavailability, or like it is highly metabolism is high, or let's suppose an IC50 value, or like all of those things, we can basically manipulate and make it a better thing so that we can have multiple hits. And based on that, it will pass the drug likeness parameters to the ADMET parameters, and followed by that we can do the docking, post-doc analysis, then validating using your simulation and this that. Yeah. Yes.
Thank you so much, sir. I
Yeah.
hope this
So
answers your question.
yeah, just
Yes,
I
yes,
would
yes, sir,
like to
please.
add in this question. So basically this scaffold hopping is a kind of wonderful this one. So earlier also we used to do as a medicinal chemist when we talk about this, because when we move from the head to lead, we need to optimize many properties, and we need to make the drug like kind of molecules. In that cases, obviously we have to come out of the some patents as well, because it's very very important. So nowadays this AI also help very effective way this scaffold hoping. There are many tools are available, or we can build up even our own algorithms to this scaffold hoping. So for example, I can say, let's say I have the kind of a pyridyl ring, and I just don't want that nitrogen in that place, right? So kind of it may be not optimized properties. It is not showing the drug properties. It has a polar nature. So what I can choose in that case, let's replace with the kind of particular pyridyl functionality with the bifunctional, or having the kind of similar properties of biosester. So what we used to do earlier, we used to synthesize these compounds one by one, then testing, and it consumes so much time, right? So but now the things are is very easy. Even we have generated one of the project we have done like that. So that project used to take the 1 to 2 years, but when we had this tool in our, and we finished this within a 10 to 15 days of this one, without without using any kind of wet lab. So this is a, kind of wonderful availability of this AI tool in this computational. So it stands in a interface of chemistry as well as computational chemistry.
Yeah, ma'am, to just
Thank
add
you.
on, just a small add-on
Yes.
to what sir
Yes,
said.
yes, sure. Sh-
Yeah. So this knowing about your molecules that sir said, I mean the nitrogen, the manganese, what is it is doing. ML works it very well now, and SHAP works with it. You must know about how SHAP is the algorithm, and that is working with with your models, with your ligands, with your nitrogen and bases, and how you can I mean as sir said, how the scaffold hopping will help you with your in the algorithms and the data set that you do. So yeah, that is the algorithm that you can just explore, right?
Thank you so much, sir and ma'am. Shri Mama Murthy, I hope this answers your question. It is wonderfully being explained and answered by all our experts. Moving over to another question. The question is from Tamila Rasu. Tamila Rasu have 4-year experience in pharma analytical field. So wants to know that how can I learn this and how this will help for my career. So I think Tamila Rasu wants to know about AI in chemistry and cheminformatics, that how one can learn this and how it is helpful for one's career.
So
Uh,
just I would like to answer. So
yes, yes,
kind
sure,
of
sir.
it's a good great to know that you are very good experience in analytical, pharmaceutical analytical field. Also, when we say about analysis, right? So we need to use some earlier is critical thinkings kind of things, and we need a lot of databases and on. We need to particularly have to chat with the data like that one to one, and it takes a lot of time. But now we have the many kind of AI kind of tools. So even we can integrate those in our regular workflow. So it can help us like that. Let's say what kind of this pharmaceutical kind of analysis. I can say this from the chemist perspective. So we have the kind of question, or we have the problem like that. We need to purify the some compounds. When we have to test the compound, we need should have the uttermost 95 plus purity kind of thing. So in that case, what we used to do, so we there is a burden on the analytical system. So, and there is no guarantee that we'll be getting the after purification, even though we have the very advanced softwares and kind of machines nowadays. But there is a kind of delays and we need to face, and sometimes we need to have compound on very urgent basis, and we have to take because time is always running out. So in that case, the AI how can help? It's a kind of many companies are come up with this one. So what it aggregates all data wherever the particular compound structure is known. So it aggregates the data, it makes the some kind of databases, and it recognizes the patterns where the what kind of purifications are required, what type of columns are required. And if we just provide the structure to the this one, it can predict at least close to that this one. So it can predict the columns which are very important, very close to that, very useful to that compound to get these purified compound. So that way directly go select that columns and using your analytical method, and get a compound within a very less time, where we used to do the so much kind of hard work. So that way it can save the kind of lot of time. Hope my answers suits your question.
Definitely, sir. Pradut sir, Snigdha ma'am, would you like to add something?
Yeah, so 4 year of experience in pharma analytical, you already have a good analyticals, with the pharma thing, with the meds, and with the personalized medicine that is coming in. I mean, all of that definitely. I mean, I'm hoping so with the pharma analytics. And um, so how this cheminformatics will help you, to go ahead. You already have a good background, I mean, right? And so on the top of it, how to use the informatics, how to use your databases, how to pre-process things, how to use your data in a right direction, how to build your models, strengthen models towards towards your prediction, to get the high accuracy, confusion matrix, all of that, to build your hypothesis, right? And then validating your hypothesis with with the basics of the cheminformatics, with with the ML as a model. All of that you'll be you'll be learning here. And with that, I mean, if that will be the amalgamation with the pharma analytics that you already have, and the cheminformatics, it'll be great for you. I mean, that will work ahead for a long time.
Thank you.
Yeah.
Thank you so much. Yeah, Pradut
Maybe.
sir, you. Yes, yes, please, sir.
Okay. So basically, when you're talking about how, like, what can help you, basically, I would suggest an hands-on internship that can help you because you are new to the informatics field. Okay. So basically, what happens is how the AI is automating the things so that you would be knowing first thing, and how to build the models. First of all, you would be getting an idea of what is R or Python. So either of the languages can help you. Okay, if you are going for Python, what is RDKit and what is Pandas, what is NumPy used for this, that. Okay, and post that, how to train a model, how you are like using the training set, test set, validation sets. Okay, and based on that, you are building a particular model. And now, when you throw a new problem at that particular machine learning algorithm, so it can be a multifaceted, what do you say, machine learning algorithm. It can be in very much depth, like your. It can be from deep learning as well, deep learning or neural network CNN based. It can be if you are using image based data, or contextual data. If you're using, you can use RNN for that matter. Okay, so all of those things you will be learning, and how it is going to help you. So it will basically catapult your career growth in the pharma sector nowadays. All of the initial screening part is, so I was just like looking at the question. Someone is asking that we still need validation of the wet lab basis. Of course, in some of the cases we might be requiring that. So the thing is, the initial virtual screening that used to take a whole lot of time in the wet lab, so that particular progression we have done, we have cut down upon the time and the cost tangent over there. And post that, the dry lab based or basically in silico based validation we can do using your simulation. And after that, whatever the screened molecules are, okay, so from like 1 million compounds, if we are boiling down to, let's suppose, 8 or 10, now it is much easier to validate those 8 or 10 compounds in the wet lab. Okay, so likewise. Thank you.
Thank you. Thank you so much, sir. I hope this answers your question. Tamla Vasu. Moving over to another question, which is from Ramu Chakrasali. That can commercially available chemical libraries used for docking purposes rather than virtually generated molecules? Sir and ma'am, uh, any of you would like to take up this question?
Working purpose rather than virtually generated models. Commercially available chemical libraries. Okay, yeah, definitely. I mean, Zinc 20 is providing that. You know, if you go to the databases, you see the databases out there. Say, for example, let's suppose Zinc 20. You know, that's that's an open source and a very well curated, organized database. If you go to Zinc 20, Rama, what you will find is there are chemicals. You know, there are chemicals that is, and all the references that is also given. Why, how, and which vendor in India or in anywhere in the world can provide you that chemical. So if you want to if you want to use chemicals, I mean, like commercially available chemical libraries that you use with your cell line cultures, your wet lab studies, you can definitely order that, or I mean, you can talk to the vendor, you can go to Zinc 20, and you can start processing that. I mean, virtually also you can start processing your building your ML, or maybe generating your results. The validation part is also important. If you're doing it experimentally versus I mean, versus the ML model, that is the best concept that you can provide to any of the journals, or any of the, like, being a reviewer. I mean, to any of the reviewers, I mean, comparing both the experimental part and the your computational part will be the best hypothesis that one can do. And Zinc 20 is one of the databases that is providing that. You can just go order that, and even if you have that in the labs, you can you can just download that. I mean, you can just have the 3D and 2D structures, and you can start your analysis. Post that, I mean, if you do docking and simulation, post that, validating it experimentally, and validating it with the help of computationally also works. I mean, doing its energy assay, right? I mean, what's the different energy assay that is providing the trajectory processes and everything. So that will make a very, very good solid base for your analysis. Yeah.
Yeah,
Yeah.
I just want to add in your this one. So yeah, it's a Jink 20 is a very nice database, as you mentioned, and we also use very good weeds. We got it from in our program. Additionally, also we can explore this enamine database. Enamine
Yeah.
also is very good.
Right.
There are billions of compounds. Also there is a kind of pubchem sites also available, because but one caution we have to take when we use this database, it should be in a very uniform way. It should be clean off, so they should not be duplicates. Otherwise, if your this data is there, obviously your outcome is impacted. That precaution we need to take care when we curate this database. Each and everything we need to have, so remove some kind of duplicacy also is a kind of one of the factor is impacting your outcome. So that's how just I want to add in your explanation.
May I also add something?
Sure, sir.
So let's suppose we are taking an example of a AI-based model like Cambeta. So Cambeta also is trained upon the libraries which are publicly available, like your Hugging Face or Bitwiz. Okay. So whenever we are building a library, of course, we need to train that particular model using some of the like freely available, what do you say, a web server only from the likes of your PubChem. It can be a drug bank, it can be a Zinc 20, it can be a Hugging Face, Bitwiz. Okay, so there are multiple of such like databases. Or if you are talking about what do you say, your plant-based phytochemical, we can the example of Impact, which is like Indian grown database. Okay, so Impact is there, and America-based Dr. Duke's database is there. Okay, Coconut database is there. Okay, so there are multiple of such a freely available database which can be used to train the model, so as we can automate the things. Okay, so that is about how the freely available database we can integrate with the AI for the training purpose, in order to build the model. The second thing is, as you are asking about the virtual screening as well as you are taking the name of docking. So docking is not only
Yes.
the technique that we use in virtual screening. First of all, okay. So first thing that we need to look at is the drug likeness. Okay, like it is following the Lipinskis or not. Okay, Pfizer, GSK, there are Weber rule, Roche rule. this, that. Okay, so we need to first do that particular analysis. If it is passing that particular thing, if the like compound is druggable enough, then we move on to the admit screening, the pharmacokinetic properties, pharmacodynamic properties we are looking at. Let's suppose I am trying to build a what do you say, like I want to come up with a lead against any of the, or I want to come up with a therapeutic intervention against any of the protein. Okay, and that particular compound is highly carcinogenic. Okay, so the pharmacodynamic parameter, that is toxicity particularly, is very high. So it makes no sense, okay, to move forward with that particular molecule and doing the docking. It might be having a good affinity, okay, but it means nothing. Okay, so that is why the initial part of the screening is also very, very important. Okay, before proceeding with the docking. So yeah, that is all from my side. Yeah, thank you.
Thank you. Thank you so much, sir and ma'am, for providing such wonderful insight on this particular question. Ramu, I hope this answers your question. Moving over to another question, which is from Ruduvan. So, Ruduvan wants to know that for an analytical chemistry master student, how does this course useful, this AI in chemistry course, and what are the opportunities in future and how to succeed in it?
Yeah, just I would like to answer. So already I
Sure.
answered the part of the answer in an earlier question by the medical. So as I explained, as you're practicing this analytical chemistry, because this is integrated part of any kind of drug discovery program, because without analysis compounds will not move forward. So it's very very important, as I explained. So here the main challenges are the analysis, how we can our reduce our time, right? It's maybe I am saying this is a hypothetical, or maybe some kind of AI tool still it is coming in a market. For example, now the main task for the main challenges bottleneck for this one. So and chemists make the compounds, but sometimes it just stuck in a kind of analysis. So that we need to have the quick analysis, even analysis done, how to interpret that data, how we can make sure that it is matching with your structure. It's very very important before giving for any kind of further biology assay, because deciding the integrity of the compound is very very important. So in that case, how we can utilize, just to answer this question. So let's say some kind of NMRs we have, some kind of LCMS we have. So there we can use AI tools. Here manually we need not to do the analysis. So once you have the set of compounds, fit that kind of structures, or kind of peaks, whatever the you are getting, and once you make the some kind of algorithm, you just need to have the kind of next level of compound. So there your time it will be saved. Also it will be less burden for the synthetic organic chemist, as well as analytical chemist. From the analytical chemist perspective, so when we talk about the kind of purification, because myself is a practice the chemistry as well, and this is the real time challenges we face. So basically the compound, when your structure grows bigger, more than 800 900 molecular above the molecule, so it's a very difficult sometimes to get the purity of the compound, and we have to struggle there. AI can help to accumulate the, or make the some kind of database. Still some companies or some kind of academics are working on this thing. This way it can be helpful. So that's I just would like to add. Hope
Thank
you
you. Thank you,
get your
sir.
answer.
Yes, definitely, sir. Producer, you would like to add something?
Yes, definitely. So as you were asking the thing about the course, so like we take like this particular course curriculum. So I would just try to convey you that we do cover deep chem, the basics of your Python and R. Okay, and then what are the libraries that are most useful for the chemist. Okay, so that we cover, and post that all of the drug discovery simulation. There's that all of the things which are required for getting started with this particular subject. So we start from scratch basically, and we move on gradually, slowly and gradually. We move on to the advanced part of the things. Okay. So this will help you in landing up to any of the good pharmaceutical companies that you see. Apart from that, if you want to go into the likes of your Schrodinger, so this that where you want to develop software, so okay, Avogadro's like Schrodinger does that. Okay, where you want to come up with the or develop softwares for them in order to pursue any of this. Like it can be docking simulation, virtual screening based tools, or likewise. Okay, so all of those things we do cover. Yes, and how it would be helpful for your future is, see, AI is future ready. So that is why if you are being trained upon this particular thing, the basic principles of AI remains the same. Okay. So whenever you are building a model, so the way you are building the model, if you want to come up with with an LLM or GNN based system, or so likewise, okay, so you can gradually build upon that, but the basis of it is itself. So you can come up with the different types of models to the likes of your neural network, CNN, this that. Okay, so that is a further part of the thing, but you need to get started first, and that is where we can help you a lot. Okay, so yeah, that's it.
Thank you. Thank you so much, sir. Moving over to another question, which is from Dayakar. So he's working as a chemist, uh, like he's a chemist working in a PSU with 10 years of experience in analytical laboratories and interested in learning AI or ML applications in chemistry. So wants to know that would this be beneficial for a career transition?
Yeah, definitely. I mean, if you're learning chemistry with the informatics, the coming up, the demand, the upcoming demand is definitely for scientists, for AI scientists, chemical scientists, informatics scientists, and the analyst, right? The informatics analyst, pre-processing. I mean, they have different pre-processing teams. They dedicatedly, they're recruiting, to maintain the pipelines, to whatnot. I mean, starting with the collection to again generating the hypothesis and all of that, right? So yeah, if you learn informatics, because you're already doing the chemical thing, the analytical lab and like the chemistry and everything. Now, because you know the core of chemistry, for you it will be easy to shift, very easy to know the informatics. You just have to work on the second part of it, the informatics. And once you know the informatics, then definitely you can go ahead with the future, because the future is the future ready. As Sir said, it's future ready, and the demand is a lot. The informatics demand is actually coming in. Yeah.
Yes. So may I add something to it as well? So
Sure, sir. Sure, sir.
yeah. Yes. So basically, when you are talking about how like you are already having a very rich experience in the analytical chemistry, okay. So how this particular AI thing can help? So of course, automation we have already talked about. Okay, but let's take some other example. Okay, so let's take an example to understand this. Let's suppose a new pandemic hits. Okay, and you don't know about the protein composition of that. So if you are going for a therapeutic intervention, this, that, whatever it is, okay, so you need to get the protein structure first. Okay, so in that particular thing, the AlphaFold can help you. Okay, so coming up with the structure of the novel protein, and after that, if you want to come up with the lead compounds which serves the purpose, or basically you want to do a drug repurposing, for instance. Okay, so for drug repurposing, you need to find the already available drug that suits that particular thing, and based on that you will be further screening out which particular drug can be utilized for this particular purpose. Okay, so in all of those things, basically your AI and ML is going to help you. Okay, so apart from that, like you can come up with the fresh libraries as like catering to your research question. So that is very important. Okay, so I might be explaining something that might not be very useful for you, but if you have the knowledge, okay, and you can build that particular tool by yourself, so you might be catering to your particular research question, so that I might not be aware of. Okay, so that is the power that the AI gives you, because you are having the potential of building out the particular tool so you can like answer your question that you have. Okay, thank you.
So just I just want to add in this one. So being as a
Sure, sir.
chemist or this one, so that could just start. I can suggest. Yes, of course, the advanced learning is very important, but as of now, because we have the many chatbots, many this one AI tools, but still we can use these chatbots and develop your prompt such a way that you can accelerate your research in a very less time, because collecting data and analyzing those things. So you may use these tools, so it definitely save your time. So that way at least you can start, and later in a further stage of this one you may think about the kind of advanced like that machine learning, and as your knowledge grows, so you can bridge your available knowledge with this kind of advanced this, and definitely you may have the better chance to have the shift in your career.
Thank you. Thank you so much, sir. Definitely, I hope this answers your question, Dayakar. Uh, I know there are a lot many questions, but due to time constraint, I won't be able to take them all. So one last question, which I would like to ask on behalf of all our attendees. Many of our attendees are not from chemistry background, like they are from biotechnology background or microbiology background, or maybe they don't have any core expertise in cheminformatics. So if somebody wants to make a career from life science background, biotechnology or microbiology or similar field, and wants to make a career in this particular field, or wants to learn AI in chemistry or cheminformatics, so I would like to know from you, sir and ma'am, that how they can start. Like, is there possible? Like, is there any job roles for them? Like, if, because obviously, if one wants to learn a particular course, they at the end they want a job in that particular field, right? So I just wanted to know, like, is there any career opportunities in this particular field, and how a person from biotechnology or microbiology background can learn about this AI in chemistry or cheminformatics?
Okay, may I?
Yeah, sure, sir. Sure, sir.
Yes. So, so basically nowadays, so there are multiple things that we handle, which are multidisciplinary. Okay, or all of the disciplines do cater to that. Just like drug discovery, if we are talking about, so cheminformatics, you would be finding it in biophysics, you would be finding it in then your like bioinformatics, computational biology, cheminformatics, of course. Okay, so likewise, there are multiple of such examples that are there. So when you are talking about how to get started, of course, anyone who is having the basic knowledge of chemistry from 12th level, so they might enroll, because as I've already stated, we start from the very basics. Okay, so very basics we start with, and I do agree with sir that you can't jump onto the AIML part of the things directly. Okay, so you should be having the what do you say, your basic proficiency of the subject first. Okay, and I would also encourage that if you are doing the stuff manually also initially, okay, if you're having that particular knowledge, that also helps a lot, because in that particular way you understand all of the principles behind it. Okay, automation is something that will help, that no doubt on that, but in order to understand the principles of all the things initially, if you are having the exposure of those particular things in the manual context also, so you know all of the steps that follows. Okay, so that is there. And apart from that, yeah, if you are from biology background, no harm in that, because in like at a particular stage or after a particular certain research stage, all of the subject tends to merge. Okay, like I have given you an example of your biophysics. If I just have to give an example of docking or simulation, for instance, okay, so the concept used over there are of physics and biophysics. The problems that we are solving, okay, are of biology, that we are seeing that how it will be bioavailable, or how the interaction will happen inside the human body. So that is biology. But all of the interaction that we see, how, what kind of bond it would be supported using, that is chemistry. So all of the things are there. Okay, so you can relate to all of them, and based on your research question, I'm again repeating the same thing, based on your research question, you can basically what do you say, your modulate your particular algorithm, and can work on that particular tangent. But In one word, yes, if you are a if you are a beginner, if you are just having the knowledge till 12th level or bachelor's level, so it is good enough to get started.
Thank you. Thank you so much, sir, for such a wonderful explanation. I know we have lot many questions, so I request all the attendees to please post your questions at cst@biotechnica.org, and our experts will be happy to answer them. So, since many of you are very much interested in learning about AI in chemistry and cheminformatics, so I'm very much excited to announce our upcoming course on AI ML in chemistry and cheminformatics, which is starting from 22nd December. Even our team has shared the link on the chat. You all can check that. So it's a 30 working days course, including project works of 3, 6, and 12 months duration. So here you are going to learn about how AI is actually used in cheminformatics and drug discovery, including from molecular representations and QSARs to GNNs, dockings, ADMET, and generative models. And also you will be working on industry-relevant tools like RDKit, DeepCam, MoleculeNet, DiffDock, AlphaFold, and real benchmarking practices like validation, leakage control, and uncertainty included. So bottom line is this particular course is built to make you competent, not just certificate rich, uh, but ready for real CADD and AI driven screening workflows. So we look forward to welcoming you and helping you take your career to the next level through this course. So as we come to the end of today's insightful session, I would like to thank our experts, Mr. Vinayak sir, Mr. Prodyot Banerjee sir, and Dr. Snigdha Tiwari ma'am for sharing your valuable insights during this Q&A session, live Q&A session, and definitely your perspectives on evolving role of AI in chemistry, real world industry applications and career pathway. The skills that you provided, the insights on tools that you provide were truly insightful and inspiring for our attendees. We deeply appreciate the time and expertise and clarity you brought to the discussion, making the session both informative and impactful. Even I would like to thank all our participants and attendees for joining the webinar and making it a great success. Your active participation, thoughtful questions, and enthusiasm truly enriched the discussion. We hope the session provided valuable insights and clarity on careers in AI-powered chemistry, cheminformatics, and drug discovery, and helped you gain a better understanding of future opportunities in this evolving field. We look forward to welcoming you to our future sessions. Until then, stay curious, keep learning, and wish you all the very best in your career journey. Thank you.
Thank you for
Thank you.
having me, and thanks to all of the speakers out there. Thank you.
Yeah,
Thank
thank
you.
you for
Thank you so
giving
much, sir.
such a wonderful opportunity. Yeah,
Thank you so much
Thank you
for
so
providing
much,
the
sir.
yeah,
opportunity.
yeah. Thank you.
Thank you.